Good afternoon, my name is Molatoli. Today I'll be presenting some preliminary results from my PhD in a talk entitled The Evaluation of ClinVar Pathogenic Variants in Patients with Developmental Disorders in South Africa. Developmental disorders are a group of conditions resulting in severe physical and cognitive impairments and usually occur at early childhood. Developmental disorders, or DD, is rather difficult to diagnose, and this is mainly because it is genetically heterogeneous, making reaching an etiological diagnosis based on clinical and phenotype information alone rather challenging. Relatively high diagnostic yields of 16 to 45% have been reported when using next-generation sequencing technologies, such as whole exome sequencing, for testing in DD patients. However, there are some limitations to the implementation of NGS, especially in African settings. These include, but are not limited to, the fact that to process the large amounts of data from these technologies, several pipelines have been developed which have been optimized using patients of non-African ancestry who present with high genetic diversity and are largely underrepresented in genetic databases. One of the aims of my PhD is to build on currently available genetic knowledge in order to develop a variant pipeline with filtering and prioritization criteria appropriate for African patients with unexplained DD, and thus subsequently identify putative disease-causing small genetic variants in these patients. Today I'll be presenting only a small subset of my results. In order to achieve this, we had 117 patient samples available. These underwent whole exome sequencing and variant calling at the Wellcome Trust Sanger Institute. We then used an automated pipeline to annotate the called variants using EnsampleVep. These were then filtered and restricted to those previously seen in other studies and annotated in ClinVar as either pathogenic or likely pathogenic, while also overlapping known DD genes. The shortlist was then submitted to our project clinical collaborators to identify clinically relevant variants. Shown here are just some representative studies results. In patient 1, following clinical phenotyping, there was a suspicion of Weger syndrome, and for this patient we identified heterozygous missense in the FOXC1 gene, which had been classified in ClinVar as pathogenic and seen in one other patient. In patient 2, there was suspicion of OFD1 syndrome, and in this patient we found a heterozygous frame shift in the OFD1 gene, which had been classified in ClinVar as pathogenic and seen in one other patient. For the last patient, there was no suspected diagnosis, but we did identify heterozygous missense in the PPP-TR1A gene and had been classified in ClinVar as pathogenic and seen in other 5 patients. The results shown here show that using the strategy of leveraging available databases such as ClinVar, we can reduce the time involved in both analysis and also the time it takes to get to diagnosis for a subset of patients. This is beneficial to our patients and their family as it allows for genetic counseling, informs on treatment and outcomes, and ends the diagnostic odyssey. We are still in the process of making clinical and phenotype assessments and will feed back any clinical findings to our patients. Thank you so much.